Welcome to our the ABIF workshop, the Introduction to Writing Macros in Fiji. I'll just go through a few introductory slides, what we're trying to do today, and we'll get going directly in Fiji. So the goal of today is essentially to kind of just teach and learn kind of a general workflow for writing macros in Fiji, with the general goal of trying to help process images and extract data from images. And so in terms of the whole your whole experiment where you generate a sample, you mount it, you get some images, and you whoops, I have to mute someone else. In terms of the whole data acquisition process, where that's it, you prepare a sample, you go to the microscope, you get some images, you do some image processing, you do some data extraction, some analysis, and eventually get to a plot that you can put either in a talk or a paper. There's a lot of stuff going on, and we're going to be sort of focusing on that sort of middle part once you've already acquired images and before making a plot. For today, you'll need Fiji, so fiji.sc, the links are in the GitHub, and we just made a few sort of sample images of DAPI, phalloidin, and mitotracker, which are available on the GitHub here, so you can download that there fairly easily. And so why do we write scripts and macros and stuff like that? Well, the idea is to really streamline the processing and data extraction, and try to help develop more robust and reliable kind of imaging assays. And the whole thing behind that is that essentially we're trying to relieve the bottleneck of tedious analysis. So I just have like a few steps kind of written verbose here on the slide. You got a bunch of images. This would be like a DAPI stain up here. And if you want to get the intensity up, what's in the nucleus in other cells, you would have to, well, open the image, adjust the contrast to see what you're doing. You would apply some convolutional filters. You would calculate a threshold somehow to sort of determine what is nucleus, what is not. And then from there, you would generate some regions of interest and then try to select those ROIs on a target image, for example, this epigenetic factor here called UHRF1, and then try to measure intensity data in each region of interest. Now that's all, that might be straightforward, that might be new to you. It is kind of a standard way to go about it. And this is fine, it's easy to do. You would have to click on all these options one after another in Fiji. And that's all doable, but then in most cases to get good quantitative data, you're going to want to repeat this process on 10 to 30 images, and then you're going to want to get replicates, and then you're going to have all your genotypes and conditions. So doing this over and over and clicking over and over is kind of tedious and can lead to human mistakes, which you'll kind of see as we get going. For me, this has been extremely useful for most of my work, just to, well, exactly help extract data from images, and this has been super reliable kind of throughout my scientific work, I guess. And I can show examples of that a little bit later if necessary. So for today, we're going to walk through writing 3 macros, which you may have seen on the GitHub. So very simple, we're going to make a composite image from a 3-channel image, which we're going to explain why in a little bit. Secondly, we're going to apply filters, threshold, and measure intensities within regions of interest, similar to what I just showed you on the last slide. And the last one, we're going to do kind of we're going to write basically a template for manual analysis, or quality control. And it might seem a little bit unintuitive to do that now. We're always talking about automation, trying to get things streamlined, but streamlining your manual analysis can be extremely valuable and extremely helpful. And so hopefully throughout all these, there's going to be a few little bits of code and little tricks that you'll be able to then apply to your own images. So we're going to be doing this on the demo images, so the DAPI, phalloidin, and mitotracker. These might not directly apply to your work, but hopefully as you sort of look into this a little bit more and consider your work a little bit more, you'll be able to kind of just tease apart these little chunks, these little lines of code, blocks of code, and apply them to your work. But that's kind of the hope over time. And so we'll be doing this in the ImageJ macro language. ImageJ, you can write in other languages in there, including Java and JavaScript, which are well supported. You can also write in Python and in R, but it's not quite as well supported. And so if you're learning this kind of from scratch, or learning this kind of early on, beginning with either programming or with image analysis, I highly recommend using ImageJ just with the macro language, unless you already know Java or JavaScript. If you're interested in learning Python and R and those things, I highly recommend it as well. Just don't do it in ImageJ. Learn Python in a proper Python environment and R in a proper R environment, because it's not quite as well supported in ImageJ, and it's somewhat frustrating to use. So the ImageJ macro language is targeted for imaging processing and analysis because it's all the commands of ImageJ into a programming language or a macro language of some sort. These are really just the written commands underlying everything that we're going to point and click on in ImageJ proper. And it's kind of a great intro to kind of scripting and programming for people, well, like me, who have a little bit more of a biology background with less hardcore programming in there. And so today we're a pretty large group. So we're currently 55. That may go up or down as things go. We'll see. So to kind of keep things moving along, we're going to ask you to post your questions in the Zoom chat. And me and my colleagues will try to get to those as soon as possible. We'll try to address them kind of in real time as they come in, and we'll have a couple of little breaks where we'll try to answer them as well. If things really aren't working, so if you have problems installing Fiji, I don't think we'll address that directly in this workshop, but all the code is available on the GitHub. And so you'll be able to basically go to the GitHub and copy paste there. And hopefully that will work out. Yeah. And we'll talk about where to copy paste it and all that as we go. Hopefully everyone has access to the GitHub, has tried it, and has downloaded the images, and we'll get going in a minute. The challenges for you beyond this workshop are essentially to figure out the processing analysis that are needed for your imaging experiments. So we're going to look at a couple of filters. We're going to say why they're useful sometimes, but you have to figure out nonetheless what you need to do. And so that's conceptually, what is the question that you're after? Is it how big is a thing? How many of a thing? What's the brightness or what's the intensity of a certain fluorescent protein? And then practically, what are the quantitative parameters that you need to extract that information? Which functions do you need, especially in Fiji? And then do you actually need to retool or rethink your acquisition settings to be able to get the information that you need? And then afterwards, it'll be up to you to figure out how to process all that data. Today, we're just going to generate a bunch of tables. And so it's up to you to figure out how to use Excel or R or Python to really go in and, essentially just plot your data or get that data into a visualizable read. So we'll go with that. There's no questions in the chat yet, I think. So we'll just get going. So please open Fiji, so you can launch Fiji, and we'll get going from there, kind of step by step. So I already have Fiji open here, and we'll kind of walk through step by step. Mostly we'll start with exercise 1, of course. We'll try exercise 1, 2, and 3 in that order, and we'll kind of see where we get to by 4 o'clock. Let's see. We'll just try to make the grid of users a little bit greater. All right. If I am going too fast, please just let me know or let people know in the chat, and we'll slow down and kind of go back. So if you have Fiji open, this is it. This is Fiji right here. It's this window. Everything you need to do is going to be pretty much contained here. And so the first things that we're going to do are essentially we're going to use 2 windows, essentially two windows in Fiji to write our macros. And the first one is very simple. It's a recorder macro. And this is kind of our special weapon or whatever to be able to do this, right? So we'll click on plugins, macros, and record. And actually, I'll just move my Fiji window to make it more visible for people on my screen. So again, plugins, macros, and record. We're currently in exercise. We'll be getting to exercise 1 if you're following along with the GitHub. And so if you click on plugins, macros, and record, you'll get this window here, which is a recorder window. And this is going to actually just record all the things that we do and kind of put out the actual command that we're going to use in our macro. And so this is where, for most of what we're going to be doing today, we're not going to be going crazy coding and writing commands from scratch or anything like that. We'll really be just sort of copy pasting commands from our recorder, and then applying little modifications as necessary to get our scripts to work. So if you have this open, then the next thing that we're open is our an actual macro editor window. So that's going to be plugins, new, and macro. I'll let that go for a little bit. So plugins, new, and macro. And that's going to open up this, this window here where it is an editor, so we can type things in here. And these will be where we're going to put in our commands. Just before continuing, we'll just need to make sure that we have the right language. So in our recorder window here, we want to make sure that our drop-down menu is set to macro and not any one of these other languages. So this is the ImageJ1 macro. And in our editor window, we want to make sure that our language is set to IJ1 macro. And this just means that the interpreter is going to read it as ImageJ1 macro. You can see that there are other languages available, but we won't be using those today. So we'll set that there. And I'll just check for, just have the chat open window just in case. Okay, so if everyone has these things open, we'll get going. We'll start with the with exercise 1. And so the goal of exercise 1 is to make composite image. And what I mean by that is that a composite image that is openable in sort of regular consumer software. So I have this folder of demo images. This is downloaded from the GitHub. If we open it, we have a bunch of, we have 15 images acquired on the microscope. And let's say we just acquired these images today. We're very excited about them. And we decide that we want to go put it directly in our PowerPoint. So we're going to say home, new slide. And we remembered for whatever reason that image 006 was really nice. And so we drag and drop. And then we see this giant black square, which is kind of disappointing. We were pretty confident that we took a good image. And so this is an example of something that doesn't work. Essentially, our images contain multiple channels. So each image file contains an image for DAPI, an image for Phalloidin, an image for MitoTracker. And things like PowerPoint and Illustrator and Chrome can't really read that properly. So we need to add at least a little bit of processing to get them into an image that we can open up on that software, and for even Windows to be able to open it and give a proper thumbnail, for example. So we're going to use Fiji. So we'll just drag and drop an image in there. We can start with the first one. So demo, DAPI, Phalloidin, MitoTracker 001. And I'll just drag and drop that here. So you can just drag and drop straight into the Fiji window, or you can use file open if that's preferable. So you should see something like this where it does look a little bit dim, but we'll get to that in a second. And the other thing that you'll notice is that there's a little slider at the bottom, and so we can go from one channel to the next. So this first one is DAPI, the second one is kind of Phalloidin, and this third one is MitoTracker, even though you might not be able to see it just yet. And I'll go back. So what we're going to do, I'll just remove this window for now, is that we're going to kind of sequentially go set the color for each one of these channels. So currently they're just displayed in black and white, and we're just going to make them each a color that makes more sense to us, and then we're going to make the composite image. So for this, we'll need something called the channels tool. So that the channels tool allows us to kind of adjust a few parameters, especially the lookup tables or these colors for these channels. So that's under Image, Color, and Channels tool. If you're a fan of shortcuts, you can do Control-Shift-Z. So Image, Color, Channels tool. And you should get this little window that pops up, Channels, with a drop-down menu that says Grayscale and a selection of different channels. So currently Channel 1 is selected. And select Channel 2, Channel 3, and that will move this slider here as well. And as I'm doing that, you'll notice that these little commands appear, and that's pretty handy because that's what we're going to use essentially for this exercise. So let's go to Channel 1. Just for fun, let's in this drop-down menu, let's set it to color and see what color we have. And it's hard to see, but essentially the color on this channel is red. Is currently red, and that's just the default of ImageJ. It does red, green, blue, and that's not what we want. So with this first channel selected, we're going to go ahead and click on blue, because we're going to set the DAPI blue. So that was in the channels tool, oops, more and blue. And then what we can do is adjust the contrast. It's very dark, it's just a display contrast issue. So for that, we're going to open our brightness and contrast window. That's under Image, Adjust, Brightness and Contrast, Control-Shift-C. And this is a window that we're going to have open pretty often when we're playing around in ImageJ. So image, adjust brightness and contrast. And then on this one here, for these images, we can just click auto, and you should be able to see your DAPI a little bit better. And now they're blue, or they were blue due to the previous. Is everyone following? Is someone completely lost or things are not working? I saw a couple of thumbs up. Thank you, those who are giving thumbs up. That's great. Oh, all good. Thank you, T-Bone. All right. So from here, we'll do the same thing with the next channels. So we'll click on channel 2. It's currently green, so it's actually the right color, but we'll set it anyways just to kind of be consistent. So we'll click on more. We'll set it to green. So that's not going to change anything. And we're also going to click auto just to adjust the contrast such that we can see it on the screen. And now you can see the actin filaments a little bit better. And then we're going to do the same thing again with channel 3. So we're going to click channel 3 here. This one we're going to set it, you can set it to red, orange, or whatever you like. These days we try to put red things in magenta just to help with for people with color blindness. So we'll select magenta, and we'll do auto as well, just to be able to see it. And so now we have all our things in color. This is still 3 different channels. So even if we save this like this, PowerPoint would not be able to read it properly. So we do have a little bit of work to do. On the one hand, we want to be able to show all 3 things at the same time. So we want to kind of these images where you see the phalloidin, the DAPI, and the mitochondria kind of all together. So to do this, in our channels tool, on this drop-down menu, we will click on Composite. So channels tool, drop-down menu, Composite. And so that should give you all 3 channels kind of together on one image. And then this is, again, this particular composite is, again, just how Fiji is displaying your images. It's still not a file that's readable by, say, PowerPoint. So we have to essentially do the last bit of processing. So you can see, and you can tell essentially because there's still there's still a slider down here, which still represent each one of the channels. They're just being displayed at the same time because we said composite. So then once you have something like this, then we can click on image, image, type, and RGB color. I'll just hang out there for a second. So image, type, and RGB color. And this should generate a new image that will be a single plane. So it will no longer have the slider underneath. So I'll just do that. We get this new image here. It is an RGB image. So within each pixel is encoded the color information. Of note, these RGB images, we never use them for analysis. We just use them for visualization, such as putting in a manuscript or in a talk or something like that. But this is convenient. We can go ahead and save this and just pretty much anywhere. So I'll just click on File, Save As, and TIFF. So File, Save As, TIFF. Make sure that it's this one. It's this new image that's selected. Every function and everything that you apply in ImageJ is going to be on the last clicked image. So if you had this one clicked in, it'll save this one. If you have your RGB image, it'll apply to this one. Whoops. And so I'll do File, Save As, TIFF. I'll just give it by default sort of adds an RGB. I'll just save it on the desktop for now. I'll say save. And then somewhere on the desktop, I should have that file. Oh, it just appeared up here. And so that's kind of convenient because then it's a good question, Jot. I'll get to that in a second. Already this TIFF file here should be better. So I can just drag it into PowerPoint and already right there, you know, oops. Microsoft is trying to be helpful. Now PowerPoint can read it, which is very, very handy. Oh yeah, that's a good question, Dina. Sure. And so now PowerPoint can read it. You can open this in Illustrator. You could throw this in a website. You'll be able to read it in email, stuff like that. And so that's kind of handy. So what we'll do is I'll get to your questions in a second because they're a little bit more theoretical, or not theoretical. You can also save as a lot of good questions here. Let's address a couple of them. Generally, we prefer, so to answer Jadavin's question, is there a reason we prefer TIFF over JPEG images for composites? Sort of. JPEGs and PNGs or JPEGs at least are much more compressed. So you do lose information. So if you do have to zoom into your image, you will see kind of compression artifacts a little bit more. And that's just because that's the algorithm of JPEG. The idea is to try to make it make images of a smaller size on your hard drive, which was much more important back in the day. So the TIFF file still maintains all the same pixel size and the same pixel distribution. It does take up more space than a JPEG, but then if for whatever reason you did really want to zoom in, you know, you can do that with fewer problems. Oops. I won't be able to find my thing anymore. There we go. So JPEG will leave some artifacts. And then the difference between the types of image in the selection menu, in the types menu, you can kind of go over this briefly. Essentially, I might not go too deep in this. Essentially, 8-bit, 16-bit, and 32-bit images are always going to be monochrome. So each pixel will only have one value. And the bit depth, be it 8, 16, or 32, that's essentially I guess the 2 to the n. So 2 to the 8 or 2 to the 16 or 2 to the 32 different values. So that means that that an 8-bit image would have values that go from 0 to 255. A 16-bit image would have 0 to 62,000, and a 32-bit image allows you to have decimals and negative values. We won't go too much into the reasons of why those exist maybe right now, but we can talk about that perhaps later or in another workshop or something like that. And is switching to RGB colors similar to flattening the image? It is similar to flattening the image in that you do, well, it's similar in that you end up with a single image, so it is sort of flat in that sense, but the color encoding maintains some information of all channels. So if we zoom in here and I just hover over a pixel somewhere here, in the main Fiji window, it's an RGB image, so there's kind of 3 values associated, one for red or one for each color. Actually, now that I think about it, I'm not going to speak. I may have made a mistake here. It's similar to flattening the image, yes, in that we do lose information, or the information is re-encoded into RGB. So yeah. Oh, the questions should be in the chat. Okay, so we'll carry on for a little bit. So we generate that image. That's kind of nice. Now, one of the things that we want to do is kind of streamline a little bit the processing. So here we did, so there's 2 things. One is that, you know, we did all that by clicking, and that was, I don't know, a dozen or 20 clicks or something on all the different menus. It'd be kind of cool to just have, if we're always doing the same thing where channel 1 is blue, channel 2 is green, and channel 3 is red for this folder of images, it'd be nice to just, you know, have one button that you click and that it all does that. So we're going to make a macro out of this. And so to do this, we're essentially going to go in our recorder folder where we have lots of information of all the things that we clicked on since the start and just kind of assemble it from there. Now, make sure you copy paste from either the recorder or like a trusted source like the GitHub. Please don't go in and type it in yourself because the syntax is critically important. Everything is case sensitive, as well as the type of quotes, the type of brackets, and whether there's a dot, whether there's a semicolon, all that. So at least at first, I recommend always getting it from either your recorder or from a macro that you know already runs. So you can also copy paste it from the GitHub. I'd suggest just to get kind of used to the mechanics of this, we kind of just copy paste it from the recorder. So what we're going to do is we're going to just basically copy paste and what we did. And of course, there's always a little bit more stuff just because, especially when I'm teaching this, I'm clicking on different things here and there. So we'll just get what's important. So step 1 is going to be to select the first channel. So I'm just going to copy paste that. So I found that here in my recorder. You may have it elsewhere. So that's stack.setChannel and in brackets 1. And then what we did to that channel, we essentially made it blue. So we changed the LUT, the lookup table, how the color is encoded to blue. And then if you remember, we also enhanced the contrast just because the default contrast was made kind of difficult to see. And that's this command here, run enhance contrast saturated 0.35. And this is the auto contrast. There's a different one for contrast reset. For today, we'll just use this one. Oops. and I'll copy paste that. So we did that. And then we did a similar thing for channel 2. And so if you did it kind of sequentially, you should have stack set channel 2. We set it to green, the green LUT, and we also enhanced the contrast. So we'll take these 3 lines, and they might be separate for you if you clicked on other things in between. And we'll paste them here. Basically, each line has to be separate. The space between doesn't really matter, so you can do it. You can put as many spaces from 0 to as many spaces as you want. It's good to add some spaces for kind of clarity, that you kind of know when things are kind of related, just like kind of paragraphs. And then we kind of have the same thing here. So stack set channel 3, run magenta, and then run enhance contrast, saturated 0.35 again. And then finally, at the end, we did 2 things. So we set the display mode to composite, just to kind of see it as a 3, kind of the 3 colors at the same time. This is helpful also just to kind of see what it looks like. Oh, a little bit of excitement. And finally, at the end, we did we made an RGB color image out of it, and so we'll paste that as well. And so if you copied all that, so this is very simple. We can then open an image again. So we'll open a different one. And then hopefully, if you have all these things in order and you have your image open and you click on run, it does all the things and it makes you a brand new image like this. We haven't yet saved it, so we would still save it manually right now, but I'm going to show you something else that we can do with this at the moment. Everybody good? So I know this for people who have done a little bit of scripting before, this is super introductory, but for those of you who it's new, this is generally kind of the workflow. We're basically trying to figure out what we want to do by pointing and clicking, and then just getting the functions from the recorder. That's most of what we're doing. And then the following exercises, we're not done with this one, but the following exercises we'll try to sort of find where that might fail or where there are little things that we can add to make our lives a little bit easier. So hopefully this worked. So that's kind of fun. Essentially this, I'll just close these things. You know, this bit of text, then you can save it somewhere. You can have it on a GitHub. You can have it in your email. You can have it as a text file. You can save as an IJM ImageJ macro. And so every time you want to do this, assuming you always have 3 channel images, then you can run this. If you have Z stacks or you have different numbers of channels, there's a little bit of modification to do, but you'll be able to figure that out. And I guess the last thing to do is that, well, you know, we have this folder of 15 images. And so it would be kind of annoying to open each image, do this, and then save. So we can also speed up that process a little bit. And so there's 2 general ways, or the main ways to do is called batch processing. So apply a function to a batch of files. And there's 2 kind of ways to do this in ImageJ. I'll show you one way now and another way in the following exercises. So for something like this, where it's very, very simple, we're always going to want these particular things, at least for an entire folder like this. Then what we're going to do is use the sort of the innate, intrinsic batch function in Fiji. And so what we're going to do is click on process and batch, and then macro. So process, batch, and macro. And you should get a window like this that appears. And for batch processing, essentially, if it's something like this, to save the macro, so we have a question about saving the macro. That's a good question. You can, it's pretty much up to you. You can do file save, and it'll ask you to save as an IJM, and that way as a .ijm file. That can be handy if you only have a couple of macros. They can be in your documents folder or on your desktop. What you can also do is copy paste and either save it in Word, or I used to use Evernote a lot, which was just kind of handy. So I have a whole mess of, you know, scripting things in here. There's stuff for Miha, who's somewhere in here, the exercises of today. I found Evernote to be useful. Or you can organize in a GitHub. It's kind of as you like, whatever you think is best. But yeah, so you can save it as a file, just to be clear. One moment, I just lost one of my windows. All right. Is it possible to do enhanced contrast manually during run? It's not easy to do things while the script is running. We will show in exercise 3 how to insert a break to allow you to either adjust the contrast or do things a little bit later. So this was a question from Nariki. Is it possible to do enhanced contrast manually during the run? Not so much. So once your thing is running, it's going to do that kind of, it's going to do this enhanced contrast command kind of in a straightforward way. We'll see if that kind of works out. So in our batch processing, so this was process, batch, and macro. If you think of batch processing, we want to apply a function to all sorts of images. And so we need to specify an input. And by input, I mean like it's really the folder where the images are stored. So for us, it's probably the demo images folder. So if you click on input, you should get a little Explorer window that opens, and then you can go pick demo images. This is the image. This is the folder that contains all those the demo images, quite simply. And then it's important to put an output. If it does save a file at the end, you generally don't want to save those output files in the same folder, just because, just to be a little bit cleaner, it's better to have like a proper output folder. So we'll click on output, and I'll just create a folder on the desktop called output exercise 1. And this is where the new files will be saved. So it's going to generate those RGB images and save it into this folder. And this is kind of good to know. You're giving an input, the files to come in, and output where to store the files that we're taking out. We're going to leave for now the output format to TIFF, although if you did have a lot of images and compression was an issue, you could put JPEG or something like that. And then this, we can jump around from one window to the next. And so I'm just going to copy paste this whole macro here. Make sure you have all the semicolons and stuff, especially the last one. And I'll just, Control C to copy and Control V to paste. And once that's in there and it looks like you have the same thing, everything's there, you've selected the demo image folder and an output, we can just click on process. And you should see your Fiji window work a little bit. And then if we go to our desktop, I should have output exercise 1. I have all these images that were created at 2:38 p.m. And so these are our TIFFs. And to see if it worked, I can actually just check directly in Windows. I can just change the view to large icons. And if they've been properly converted, you'll be able to see them in your folder. And so this is surprisingly useful. One, we just went over batch processing. You can apply a function, whatever it is, to a whole folder of images. And I do recommend that you do some visualization-based processing on your images, just because it'll make your life easier down the road. So you could put this output folder in the original folder, and it's just easier in a month or two when you think, I did this staining, 2 months ago, 6 months ago, something like that. I remember there was one image with a crazy cell that took up the whole field of view. Which one was it? And if you just have these little thumbnails here, it's super handy to say, oh, it was image number 13. And then you can, you can either go back to that to do quantification, you can put it in your talk or something like that. And just kind of seeing all the images also just kind of gets you a little bit closer to your imaging data. So it is kind of helpful, as opposed to just having, the demo folder, which, if I just change the view here, you would have to open each one of these separately and kind of have a look through Fiji, which is important to do, but also kind of a little bit more tedious. So having this here, you can already just see everything. If you had time-lapse movies, you can make AVI movie files that can be read by Windows and PowerPoint and whatnot. So it's just kind of handy to have these kind of quickly to be able to say, oh yeah, cell number 009 or image number 009 was really nice and demonstrates the type of cell that I'm interested in or something like that. Okay, I think that's it for exercise 1. I guess a couple of little things to point out. We can do a little bonus thing. So right now we copy pasted from the recorder. This works because our images have 3 channels. Where it might fail is if you have images that do not have 3 channels, because it will not be able to find, for example, channel 2. If you have a single channel image and you say, hey, set channel to number 2, it will throw an error that it can't find channel 2. So this is where you might have to do a little bit of adjustment of, if you have other images that only have 2 channels, well, you could either delete this entirely, the channel 3 part, or kind of perhaps a more nifty thing to do is to comment it out. And so a useful feature in programming, in most programming languages, is a comment feature. And in the ImageJ macro function, ImageJ macro language, it's 2 slashes, 2 forward slashes, so the divide by thing. You'll notice that it should make your, you can put it at the beginning of each line, it should make that whole line green and italic. And that just tells the system to ignore this little bit of code. And so that can be useful just to not delete it, just to make sure everything's there in case something was needed later on. And that also allows us to do kind of useful things just to give us information. So we can put a few spaces at the top. And on the first line, we can say, you know, macro to generate composite images from 3 channel. And then you can give as much detail as you possibly can, just because for something like this, it's fairly simple. Once you start writing things that have more than 20, 30, or 100 lines, then it's kind of hard to figure out the entire, exactly what each thing is doing. So it can be helpful to just put comments here and there. So, you know, first channel is set to blue. You know, you can say second channel, etc. And you can put your name if you like. Emoji too, why not? You want to be friendly with yourself when you come back in 6 months and wonder what you did. And so this is really handy because then you can also go into for each kind of paragraph of code that you have, you can go by and kind of just add some information such that later on it makes more sense. Again, this particular script is a little bit relatively easy to follow, but once it does get a little bit more convoluted and you have to do a little bit of pirouetting to get the values that you want, then it's kind of helpful to add some information there. Okay. Are there any further questions, or did things not work for people? Right. It looks okay for now. So I hope this was helpful. This was exercise 1. Hopefully you can kind of get an idea, a feel for how these things work out. Okay, so we'll move on to exercise 2. I'm just for clarity, I'll just go ahead and delete everything in my recorder just to make it a little bit more clear. I'll move this here. And I will we'll create a new macro tab here. So that's just in the macro editor window, click on File and New. And then again, we just want to make sure the language is set properly. So right now it's not set to any language, which is not great. So we want to set it to IJ1 macro. If I just go back to the composite macro that we made, you'll notice that there is kind of markup and different colors based on what's there. So if you don't have a language selected, it'll all, oops, I didn't like that. Can't go back. Yeah, we'll do that again. If you don't have a macro language selected, it will all be just appear in black text. And then the sort of color code can help you sort of figure out what's going on, which is kind of helpful. So most of the time, functions and commands are going to be in yellow, brackets are going to be in red, integers are going to be in purple. These are strings, so this is just bits of text. This is how functions are referred to using the run command. Things like file names and little bits of text where you might write like, oh, the width of my image is, that will be a string as well. That's going to all appear in pink, and your comments are in green, essentially. Oh, right, and on Macs, that's a good question, and thanks for answering, Elka. On a Mac, usually your all your menus, be it for the main Fiji bar or for the macro window, are going to be at the top bar of your screen, which also means that you have to click on the main Fiji window to get file, image, process, etc., or on the macro window to get file, edit, language, and all that. Yeah. Great. Okay, so we have a new window here. The language is set to IJ1 macro. We'll just put some comments down, so 2 slashes and exercise 2. And we're going to, this is going to be, you know, run filters. We'll discuss in a second. Threshold DAPI channel. And get intensity from actin channels, let's say. We're not going to batch process this directly, but we're going to talk about it in a little bit. So we'll kind of walk through this. We'll walk through this step by step as we did for the previous ones. Here we're going to access a few more functions, so really the filters, and you'll kind of get an idea of the mechanics of this. We're going to run into, I believe, 2 issues that we have to solve. By issues, I mean things that that we'll have to add in to the macro to make it run that will not be directly, that we can't directly pull from our recorder window, and those will become kind of clear as we go along. So we'll open an image for this particular exercise. Just go back to my details. Let me go back to the large. Actually, I'll consult the nice folder of images that we have. Let's find an image with multiple nuclei. Oops, that was not right. Where's my output folder? So let's choose image 11, but of the raw images. So in the demo image folder, let's go choose image 11. So I'll just drag and drop it here. And so we have DAPI fluorided micro tracker 11. If you want, you can adjust the contrast on each one. So you can click auto just to see a little bit better. I'll just do this kind of by hand. Just kind of see these things a little bit better. We'll leave it kind of in grayscale mode, just because it's a little bit easier to work with in this type of situation, in a single channel situation. Our recorder, I'll just kind of hide it somewhere up here. We don't really need it just yet. And it just kind of runs in the background and we're all fine. So the goal is to, this is exercise 2, different goal. We decided we want to do some analysis on this and get some quantitative data. So we're actually going to make some measurements and try to do it as automated as possible. This is similar to the exercise that we did in the previous workshop, I guess well over a year ago. So we'll do it again. Different images this time, I guess. So so what we want to do is get the shape of these nuclei, and we could do this manually using the freehand selection by going in and kind of drawing around a nucleus, and just to generate kind of a shape. And then within this shape, we can then go to our second channel, and then we could measure the intensity within the nucleus of, say, actin. And I just hit measure, we'll get to that in a second, and here we get some information. Perhaps before doing this, we'll set our measurements appropriately. I always forget. So if we click on Analyze and Set Measurements, so Analyze and Set Measurements. So in this Set Measurements window, you can choose the measurements that will appear in the results table. And this is important to set before starting, just because if you do all your measurements and you end up with just the area and something else, maybe you want more information and you have to do your analysis again, or at least run your macro again. So I typically have a few things there just because I think it's useful. And in the downstream processing that I run either in Python or in R or MATLAB, it's fine if there's extra information that I don't need for that particular experiment. So things that are useful to have are the area. So that's going to be the surface area of the measurement. That might be the whole image, or that could just be the region of interest. So in this case, it will be like the nucleus. The mean intensity value is good. That's literally the kind of gives you a rough readout of the intensity within that area. Standard deviation is often helpful, and I often use it as an actual measurement of clustering, which we won't get into details now, but gives you an idea of kind of the range of the data around the mean. So if it has a low standard deviation, that you could imagine the intensity being very sort of uniform. If it has a high standard deviation, that means bright spots and dim spots within your object. I like having the min-max gray value clicked in. This is kind of handy, especially if you do get weird values. It's kind of quality control and sanity checks. The centroid is the XY positions of your region of interest. So if we did create a shape like this, we would get the XY position of the spot right in the center. And that can be good for troubleshooting, especially a month later. If you do see a weird value, you can go back to your image and say, oh, there was something weird at position 600, 422 or something. I use the integrated density a fair bit. That's just the total sum of intensity. The median, so that's the median intensity of the inside here. The median can often be useful compared to the mean. If you think of those two statistics, the mean is extremely subject and sensitive to outliers. So if you do have super bright pixels, noise, stuff like that, depending on your acquisition modes, sometimes the median is a better reflection of the actual intensities within your sample. And finally, just for good kind of documentation, the stack position. So are you on the first channel, the second channel, third channel, different slices, time points, stuff like that. And the label that gives the actual file name and the region of interest if someone was there. So this is also just for documentation. Again, 6 months down the road, if you see a weird value, you can go find the file, the stack position, and the XY coordinate of your measurement, and you can determine, oh, it didn't work properly, or oh no, that's just biology. Biology is weird, and kind of go from there. So I do recommend these ones. You might need others depending on what you're doing. So perhaps you need the perimeter. Ferret's diameter gives you kind of the whole length. I think it's the greatest length within the object, and there's a few other measurements there that might be useful. These ones are pretty common for me, so I'll select that. I'll say okay. When you do that in Fiji, it kind of saves it. So the next time you open Fiji, it's there. However, you still get the command here. So you can go ahead and copy paste that into the macro window right now. So that's run set measurements, and we'll just put that at the top. So that just sets it, especially if you go use Fiji on a different computer where those are not saved, then you know that it's going to run properly. Okay. So as I said, we can manually draw out these nuclei, but that's a little bit time consuming. Sometimes you have to do it if your images are relatively low signal to noise. But in this case, we're going to apply a few commonly used filters to be able to try to threshold them in an automated fashion. So I have a question. Could you explain the difference between the MGV and the integrated density? What was MGV? Oops. Did I miss something there? Oh, mean gray value. Oh, well, I keep clicking on the wrong thing, sorry. The mean gray value is literally the average intensity. So, so if you take something like this and you look at each of the intensity of each pixels, it just gets the mean value. So if you think of this particular DAPI stain nucleus here, we have values around 26 and 31 and 33 and this and that. So the mean would be somewhere around, 30 or 25, the gray value. Whereas the integrated density is really the sum of the value of each pixel. So it'll be, how many, well, each pixel here you're adding them together. So this will probably be in the tens of thousands or something. So in terms of interpreting that, if you're this is really tenuous, and be very, very careful about how you do this. The mean is a little bit more like the concentration to a certain amount, the concentration of fluorophore, sort of, and the integrated density is like the abundance, more akin to how many fluorophores are there. Again, you can't, it's difficult to make these direct parallels, and you have to be very, very careful when you try to do this. You have to have a lot of controls to be able to make these types of claims. But just generally speaking, it's total amount versus average amount. Yeah. All right. So let's move along. So generally, when we're, let's make this a little bit bigger, we want to get these shapes. And so for whenever we're doing functions that do modify the image a fair bit, we don't want to do it on the raw image, just because we don't want to accidentally overwrite our raw image. So what we're going to do is duplicate just the first slice, because for now we'll just be working on the DAPI. So we'll say image and duplicate. So that's Control-Shift-D, image duplicate. And before clicking OK, we want to uncheck duplicate hyperstack. We really just want that DAPI image. We don't need the phalloidin and the mito tracker. So if we unclick duplicate hyperstack and our DAPI is displayed here, we'll get just the DAPI image. So again, if you have the DAPI in your channel select here, say image duplicate, uncheck duplicate hyperstack, and say OK, and you'll get that single channel image. I'll just close the second one I duplicated. And this is handy. We usually do this just because now we can kind of mess around with it. And if we do something silly, we can just, you know, delete it and start over again. So we don't risk messing up our raw data. So then for this, what we want to do is apply a threshold. And applying a threshold simply means choosing a pixel intensity value above which is our objects of interest, our signal, and below which is just background. So there are multiple ways to do this. Usually we do a little bit of processing on the image first to kind of help out with that. So I'll just kind of show you real quick. If I crank the contrast substantially, you'll see that if we zoom in, there's a little bit of noise. So by noise, I just mean kind of these slightly brighter pixels over the background. They're kind of just spotted here and there. In the particular case of these images, it's not so important, but you may very well end up with images that do have lower signal to noise, so your signal is less bright compared to this noise. And so it can be helpful to sometimes get rid of this, especially in confocal images that can have this sort of shot noise a little bit more. And so one way to do that is by applying something called a convolutional filter. I won't go into detail too much, but a convolutional filter, what it does is that it'll go over your whole image, and for each pixel, it's going to make a little square, a little circle around it, and apply a transformation to the central pixel based on its surrounding. So, and it's going to do that for each pixel. So it'll do that one, then it'll move over a little bit and do the next one, next one, next one, next one, next one, and do that over the whole image. We're of course not going to program that. There's a function that does that automatically for us. And there are different functions that we can use. One that is kind of handy for denoising is a median filter. And that is maybe more intuitive than what you'd expect. If we have a pixel, say this one here, and we want to apply a median filter, we can say, well, okay, instead of having this pixel here, which is a little bit brighter than its surroundings, why don't we just give it the value of the median of its surroundings? And usually that's an okay assumption to make, just to reduce noise a little bit, to reduce noise in the context of processing to be able to generate objects. And so to do this, I'll just make sure nothing is selected. This is going to be process, filters, and median. So process, filters, and median. And then from there, we're going to give it a very small radius. Essentially, we're going to give it a radius of 1 pixel, because that's all we need. And that just means that each pixel is going to be, the value of each pixel is going to be replaced by the median value of its surroundings, quite simply. And that's generally going to get rid of little fuzzy noise like that. I'll say okay. And you can already see that a bunch of little spots have, you know, disappeared. Some remain because they were attached to something else, but it helps a little bit. And so that's kind of a common procedure to suppress noise when we're trying to get objects and regions of interest, stuff like that. I'll point out that it was not necessary for these images, but I just kind of want to sort of show you that this is a very commonly used filter. Perhaps before moving forward, we'll just we'll copy paste some of our commands. So what we did for our image, essentially we duplicated it. So if we go back to the start, and this is just to kind of get it in while it's still somewhat fresh in our minds, we first duplicated the first channel. So I'll say run duplicate. I might even add, just to be safe, a little stack set channel to 1, oops, from our previous macro, and put that on top. So that way, if we do open the image and we do adjust the contrast and we're on the mitochondria channel, it will go back to the DAPI channel. And then when we duplicated, when we ran the duplicate function, it created this new window here. And that new window becomes the active window. So whatever function that we apply next, it will apply it to this duplicate. So that's cool. We don't have to really worry about selecting that image or anything like that. So then after that, what we did was apply a median filter to try to suppress the noise a little bit. So we'll just find in our recorder, run median, and that's going to run the median filter on this. I also have the Gaussian blur in the GitHub. We'll skip that for now because it's not so critical. And you can try it on your own, see how it modulates the image a little bit. So now our macro is mostly up to date. We did our median filter. And now we can start, we can think about putting setting a threshold. So a threshold just means finding an intensity value that, again, that below the intensity value is going to be our background and above it's going to be our objects of interest. So to look into that a little bit, we'll click on Image, Adjust, and Threshold. So Image, Adjust, Threshold. And we'll get this window that pops up, and your nuclei might appear red a little bit. Don't worry about that just yet. So in this thresholding window, basically what we're saying, the pixels in red are the pixels that would be selected or kept or above threshold given these current settings. In the threshold window, you have two sliders, one that determines the lower threshold, so the minimum value that's needed to select those pixels, and so you can play with that a little bit. And you also have another slider here that's the maximum value. So you could select kind of a lower limit and an upper limit if you wanted. So you could bring this in. Suppose you really just wanted the edges of your nuclei for whatever reason. So you can do that as well. So you're just selecting a certain band of pixel intensity values. Those pixel intensity values are given here. I'll just stretch this out entirely. And so we can sort of do this manually. So you could go in for each image and say, I think a threshold of 9 is good for this one, and maybe the next image could be 10 or 8, or maybe it's also just 9. However, typically what we try to do is get the computer to calculate that for us, simply because then it's a little bit more robust and doesn't require the human intervention or anything like that. And so commonly used, there are different algorithms, different ways to figure out what the threshold value is in an automatic way. We have those in this first drop-down menu here, where it probably says default. And you can have a look. You can a couple of them. These are just algorithms that are over time. There's one very simple one. It's called mean. And so that just takes the mean intensity of the image, and everything above that average intensity is thresholded. Everything below is kind of discarded. Yeah, getting a little bit of fuzz here. There we go. And so the mean intensity doesn't work so well in these images. An algorithm that's often used is called Otsu. And this is regularly used for fluorescence images, especially for bright particles on a dim background. And it's a little bit more responsive, essentially. It seems to work pretty well with a lot of images. And you can see what would happen if we run it. It looks pretty good. It looks like the nuclei. We can say reset and then auto, and it seems to get those shapes pretty well. Otsu, I'm not exactly certain of the internal workings of the algorithm, but essentially I think it tries to find a population of background intensities and a population of like signal intensities, and tries to find a trough between those two populations somehow. So, and then if you use Otsu, that's kind of a standard algorithm. So you can say in your methods like, oh, we used Otsu to generate the threshold, again, to choose that value above which we're going to keep signal. So these particular images work well. Okay, sounds good. There's a couple of questions. The threshold slider window doesn't appear when I click adjust threshold. Well, we'll give it a try. It might be hidden behind another window. Yeah, so you can try it again, adjust threshold, or you can try to click on the and if you click on like the finder button at the bottom, and you have all the other windows that pop up. It's a little bit strange that it's not there. What we can do is, at least for the purpose of writing this macro, you could get the script from the GitHub, because it'll be just 2 lines of commands, and I'm assuming the threshold still exists in that version of ImageJ. The sliders might look different, yeah, on the Mac. Okay, so getting back to this, we chose Otsu, and then we figured that that's pretty good, and it will be pretty good for these purposes. And so we'll just click auto. That looks nice. And so we'll just click apply. You'll notice this changes the image substantially. You should get an image that looks kind of like this. The colors might be inverted. We'll see how it goes. And this is no longer an intensity image. This is really a binary image. And by binary means that the pixels have one of two values, either 0, which is everything in white here on my screen, which is just background, and a maximum value of some sort. So in this case, it's 255. Doesn't really matter. All these objects are going to have a value. Each pixel in these objects can have a value of 255. And so when you just have those two categories, 0 and 1 or 0 and 255, it's very easy for the computer at this point to say, oh, I'm just going to keep these objects here, rather than try to make a decision of, where does the nucleus start and end. So let's just copy these commands a little bit before continuing. We did set auto threshold Otsu dark. So hopefully you have that in there. And then we did run convert to mask. So that was just what we did with this threshold window. Set auto threshold Otsu dark. It has to be written like this. It's case sensitive and all that. So as much as possible, try to copy it. And it should be accessible in the GitHub as well. And then run convert to mask to generate this binary image. Once you have this, this is a binary image. These are masks. We can basically ask the computer to give us the information, the spatial information, or make an object out of these things. So one way to do it is to use the analyze particles function. And so that's under analyze particles. Analyze particles. And here we have a few options to look at. You'll probably have something that looks a little bit more like this. So for these particles, we can, there's a few things that we can play with. Essentially, analyze particles, it needs a binary image with, stuff with either 0 or 255 as values. And then it'll create an object for each kind of separate object. So each group of 255 pixels that's not touching anything else, that's surrounded by 0 pixels. Now with that, we can apply a few filters. So notably, we can put a size filter. So for example, if we know the size of our nuclei, or if we know that there might be debris of a certain size, we can try to just immediately get rid of it. So these nuclei are about 100 by 100 pixels. So that means that they're a total of 100 times 100, which is 10,000. So we can put a minimum size just because we might have debris that's much smaller than that. I'm going to put a minimum size of 2,500. And that means any object that's about 50 by 50 pixels will be ignored. And we'll just go till infinity. You could also put an upper limit as well to get rid of large objects, especially if you have nuclei that are close together and you want to throw out things that might be that weren't thresholded properly. And we're going to put it in pixel units. For a lot of this analysis stuff, it's easier to think in pixel units rather than in microns. However, it is important to know to get a good understanding of the absolute units of the things you're measuring. So usually these nuclei are around 10 to 15 microns, let's say. But for now, we're working in pixels. So we'll set a filter from 2,500 to infinity. Then we can choose the circularity. Circularity is a calculation simply that that gives you an idea of, well, the circularity of an object, where you end up with a value of 1 if it's a perfect circle, and a value closer to 0 if it's very, very distant from a perfect circle. This can be really handy if you're segmenting if you're segmenting whole cells or even nuclei, simply because things like mitotic cells are very, very close to being perfectly round, and things like dead cells are also very close to being perfectly round. So you can actually filter those either to keep them or discard them from your analysis using the circularity tool. And I said mitotic cells and dead cells. This is more like in cultured cells essentially, but this can be helpful as well. For show, we'll say show nothing. We don't want anything else to appear. And then in the boxes, we'll click add to manager. This means add to the ROI manager, the region of interest manager. Essentially, each one of these objects will become a region of interest that we can then manipulate. And just as good practice, we'll say exclude on edges. So that would mean if we had a nucleus that was right on the border of our image, that was touching the edge, we would ignore it. And that's generally good practice simply because, well, you don't have an image of that whole cell. So maybe there's something weird happening just outside the cell and you can't see it. And every once in a while, depending on older systems, sometimes the last column of pixels can be a little bit iffy from the data transfer on your detector. So sometimes it is worth getting rid of that as well. But essentially we get rid of stuff that we don't have all the information for because it's not worth it. So essentially, we're just going to keep add to manager and exclude on edges, and we're going to say OK. And we're going to get these, should get something like this, where you see, you should get something like this, where you see the different values kind of, these different numbers for each one of these objects. There should be like a little bit of a yellow outline to these things. And you can toggle them on or off as you please. Notably, you probably got this little window that popped up that's called the ROI Manager. This is super useful. This is going to be your friend for any sort of object detection type thing, whether you do your thresholding in Fiji or you use some sort of deep learning-based neural network thing. You're still going to work with this ROI Manager a lot. It's super cool, and we'll get to it in a second. We'll just copy the functions that we did. So we'll go back to a recorder, and we'll just take this analyze particles line. So run analyze particles. And you can kind of see just in some of these functions that we're writing, there is a certain structure to the arguments. So our median filter, for example, we gave it a radius of 1. For this particular function, it has to be input like this. So as a string, lowercase radius equals 1. And for the analyzed particles, all those things that we put in sort of gets stretched out into this string, this bit of text here, which means we have to leave it like that. We can't just say, you know, add to the manager, like that. That won't work. So we sort of really leave it in the way that it's output, because the commands are expecting that. And just to make sure I didn't do anything silly with that little example, I'll just copy paste it again. It looks okay. Anyways. All right. And so what this did is that it stored information, basically the spatial information of all these shapes. So it stored each one of these shapes into this little thing here, which we can click on. And that means we can go back to our raw image. So I'm going to click on the raw image here, and then we can apply these ROIs kind of as we please. And so if we want to measure the intensity of the actin filaments under the nuclei, then we can just go to our second channel, and we can just make measurements here. So to do that, you would click on one of the regions of interest in our ROI Manager, and then to measure, you can either click measure here, or you can hit the button M, the letter M on your keyboard, and you should get a little table that appears. Then we'll go to our ROI Manager, we'll click on the next one, we'll hit M, we'll go on the next one, hit M, next one, hit M, and next one, hit M. Okay, so now all we have to do is copy paste those things. And you could probably start getting the impression that something weird is going to happen. So what did we do after analyzing the particles? And that this ROI Manager pops up, we clicked back on our raw image, the original image. So let's go in our recorder and find that. And so after all my select objects here, or before all that, I have this command called select window. So for now, I'm going to copy paste that. And you might notice something a little bit problematic with this. It says select window demo dapi phalloidin mitotracker 011.tif. You can probably kind of guess that this function select window just looks for a window with a certain title to it, and it's going to look for this particular title. So that's not great. This is what's called hard-coded, which means that the value is what it is and is not mutable. And this is not really good. If we leave it like this, it will work for this particular image, so demo dapi phalloidin mitotracker 11, but then the second we have another image, say number 12 or number 13 or number 2, it won't work because there will be no window that's called 011. So this is where we have to start getting a little bit more clever and start harnessing a little bit some of the computational tools or some of the programming tools that we generally use. And so there's multiple ways to do this. We could rename this. We could rename our raw image to something and always reference that. Typically, I avoid renaming things just because it's kind of nice to have the full file name, to be able to use the full file name in our measurements. That way we end up with the file name, for example, in our results. And if we create a new file, it still has the same 011, which is kind of useful. So it's not usually handy to use the rename function. And another way would be to essentially use variables. So we haven't really used any yet. And so a variable, if you remember from your algebra, it's just a symbol that that contains something. So from algebra, it was usually a number. And in programming, it's to contain anything like either a number, a bit of text, or in the case of ImageJ, you can also store an image. And so a variable can be given any name. We can call something my variable, and it could be equal to 5, or it could be called my variable, well, poorly spelled, equals hello world, if we wanted to reference that. Or it can also be an image. And so for something like that, we'll kind of have to type it as one. We'll give it a name. In this case, we'll call it raw image, just because then it's clear to see what it is. You want to try to give your variables easy to reference name. We'll say equals, so we're going to assign it. And this is a command here that we'll have to get that doesn't appear in the recorder, but that exists in Fiji. It's called get image ID. And so what you can do, if you have Fiji recently, you can type get image all in one word. It's lowercase g, uppercase I. And then you should get the autocomplete here for get image ID, 2 brackets, and a semicolon, and double click on that. And so that is going to save, I'll delete these my variable things. We created this this variable at the top of our script, well, after set measurements, because essentially what we're going to do is that we're going to open an image, and then we're going to want to call it immediately, that image, whatever it is, whether it's 11 or 12 or 13 or 14, it's going to be known in the system as raw image. And so that means then we can do all our other activities, so set the channel to 1, duplicate it, do our filtering and all that stuff. And then instead of set select window, I'll just comment it out. We're going to use a different one called select image. And so if you type select image, it should appear again in your autocomplete. And the ID is just telling you that you have to put an image ID there. And so we're going to replace that with raw image. So we'll just copy paste raw image here. If you're worried about the syntax and getting it properly, you can follow along on the screen. So it should be, things should be yellow and red and this and that. Alternatively, if you go on the GitHub, there's exercise 2, an exercise 2 file, and then some of this information is there. So for example, get image ID is would be on line 19, and select image would be on line 38. Sorry, I can show it to you here. So if we go to the GitHub, exercise 2, and then we scroll down a little bit, I added more comments here. Don't worry about that too much. On line 19, you can go get raw image equals get image ID. And then on line 20, line 38, sorry, you can get select raw image. And of course, if you can use the autocomplete from Fiji, that's helpful. It's a little bit finicky because you have to sort of start over sometimes, but it should work out. So this is handy. Using this type of thing, we can then switch between our images as we please. We could create another variable for our duplicated image if we needed to go back and forth. So right after duplicating, we could say, you know, duplicate image equals get image ID. It's not necessary in this case because we're probably not going to refer back to it. We already got the information we needed from that duplicated image. All right, and so does this make sense to people? Is there something wildly confusing? I got a couple of thumbs up. This is good. Thank you, thumbs up, people. So this is handy. It allows us to switch. So it allows us to do some of these mouse things. It's one of those commands that won't easily appear. So there's a question. If I was only writing code without following the recorder, how would I exit from the duplicated image? So you have a lot of options. In this case, so what we're doing now, when we're saying select image, raw image, we are effectively leaving that image, right? The image that we're going to is this raw image. The duplicated image is still somewhere in the background, but it's no longer it's no longer going to be active or used for analysis purposes. We would have to, if we wanted to go back to it, we would have had to create a variable here. So right after duplicate to be able to switch back and forth. If you really want to close it, there is a close function quite simply that we'll get to a little bit later. Yeah, so if you needed to close it for whatever reason, or if you're doing like a batch processing, you want to close the images as they appear, there's a close and a close all function. And then the next question is, in other words, follow-up analyses will automatically be conducted on the original image. This will depend on what you're doing. So what we're doing today is that we use that duplicate image to get those shapes. And then we're going to use those shapes to measure the raw intensities on our original image. In other situations, though, perhaps in other analysis situations, perhaps you'd want to do some processing on those images as well. So maybe you would want to apply a median filter to suppress noise to then get a better indication. So it's not always that you're going to be applying back to the original image, but one thing to know is that this duplicate image now no longer has any intensity information to it. All it has is 0s and 255s. So once you generate these regions of interest, there's really not much more information that you can get from it. So in most cases, you would go back to your original image, or go back to your original image, duplicate, say, the actin channel, do a transformation of some sort, and then measure there. But this duplicate image is now a binary image that we extracted we squeezed out all the information that we can get from it. Yeah, that's a good question. Right. So now we selected our raw image, and now let's have a look at I'll just redo some of these measurements. So I'll close this, the results table. If we go here, what we did was select something and say measure. Oops. Select the other one, say measure. Next one, say measure, measure, measure. And in our recorder, we get something like this. So now you can kind of guess that this is a little bit weird for a variety of reasons. If I just copy paste this, this will work on this particular image because there is this number of regions of interest. But let's just unpack this a little bit. So essentially for each region of interest, it was selected, and then we ran a measurement. So if we look at this first one here, it says ROI manager select. So that's the command to select one of these regions of interest. And then the number 0 is the position within the list of ROIs. So if you consider this list here, it's at the 0th position, which is kind of weird. Essentially, whenever you have a list of things in ImageJ, and it's the same in Python, the first object in that list is always going to be at the 0 position. So what that means is that for this collection of regions of interest, I have 1, 2, 3, 4, 5. There are 5 nuclei. This one here is at position 0. This one at position 1, 2, 3, and 4. So it goes from 0 to 4. So that's a little bit weird, but that's just kind of the way it's encoded. And there's a few reasons for that kind of downstream in the programming world for that. So what does that mean? So we selected the 0 one, we did measure, we selected the first, we measured, blah, blah, blah. So this is fine. This would work on this particular image, but there's 2 problems here. One is not so bad, but not great. If you have an image with more nuclei, so let's say you have an image with 10 nuclei, then, and we just had this code, then it would only image the first 5, or it would only, measure the first 5. So that's not great. It would not get the information from the next 5 because we didn't tell it, hey, select number 5, number 6, number 7. So that's not great because we're losing information, or we're not getting that data, which is not ideal. And on the other hand, the worst case scenario is if you have an image that has fewer than 5 images, fewer than 5 nuclei, then let's say it only has 2, then it's going to go to, select 0, it's going to measure, that's fine. Select 1, that's fine. But then select number 2, which is essentially the third, it's going to throw an error because it'll say, hey, there's no third object. I can't do this. So this is not good. We don't want it to fail based on the number of objects there, and we don't want to have a fixed recording of number of objects. So we need to figure out something for this. And this, for this type of work, this is a standard programming expression that will write and will unpack. And it might still be a little bit unintuitive at first, but you'll gain an understanding of it a little bit. Essentially, we want two things. We want to, for any image, regardless of the number of regions of interest, we want to get that number of regions of interest and then apply the function measure to each region of interest regardless of the number. So regardless whether there's 5 of them, 2 of them, 0 of them, 100 of them, we just want to apply it to everything. So for this is going to be an iterative process. We want to do the same thing iteratively. So on ROI1, ROI2, ROI3, up to n ROIs. And so for this, we're going to write something called a for loop, which we'll get to in a minute, and we'll unpack. So first, what we'll want to do is just get the number, get the number of ROIs, of regions of interest, or nuclei, in this image. So how many of these particles did we find? So there are 5 in this image. Another image, maybe there's 2, there's 10, kind of whatever. And so for this, we're going to create a variable, and we're just going to call it num ROI, something like that. And there's a function in ImageJ to get the number of ROIs. And so that's ROI manager. And so you can say ROI manager, and you can select count. So in brackets with quotations, ROI manager count. So once you have that, and again, if you have trouble finding it from the autocomplete, which it doesn't always work all that well, you can get it from the GitHub in exercise 2 at line 41. There's num ROI manager count. And I'm just going to delete this. And now we're going to write our for loop, and this is a little bit weird. So we're going to just type puts in some comments here, write a for loop. And the syntax is kind of important, and we'll unpack it. So we type for, and then we get the syntax in our autofill, and we'll unpack it. And this is a loop, so there's a curly brace. I'll hit enter. And within our curly brace, I'll just, for the time being, put a little bit of code. So we'll say ROI manager select and run measure. And what we'll do, we'll just fill it up completely how it's supposed to look. And then we'll take a little break. And then we'll unpack how this works essentially. So right now we just kind of auto-filled this. So what we want is for I equals 0 till I is lesser than the number of ROIs. So num ROI, this variable that we created earlier. And I++, this is an iteration of +1, which we'll discuss. And then we're going to say the commands that we want to repeat over and over and over, which is going to be ROI manager, select, and in this case, not 0. We don't want to select 0 all the time. We're going to want to select I. That's going to be the iteration. And then we're going to run the measure function. So make sure you have this written down, or again, copy paste it from the GitHub. It's the last few lines from exercise 2. Essentially, num ROI, we're creating it here and we're calling it a little bit later. We changed ROI manager is going to be select and then I. Again, please try to copy paste this as much as possible. You can have a look at kind of the colors, and there should be another closing curly brace at the end of all this. And perhaps since we have been going for 90 minutes straight, we'll take a little break. I'll stick around if there are questions, and we'll unpack this kind of element by element with visual support in, let's say, 5 minutes. Okay. All right, so we'll get started pretty soon. Some good discussion as to where some of these symbols are on keyboards with different language settings. I I kind of always oscillate between having my keyboard in English or in French, and so, you know, sometimes you try to do a slash and it's the accent aigu, and it always kind of looks silly. I'm sure a few of you have analogous problems with that as well. All right, so so we were going to talk about this for loop. As Evie pointed out, yes, we should probably select our channel. We're going to select it inside the loop. Essentially, so in this case, let's say we want to measure the intensity of actin under the nucleus. So we have our regions of interest from the DAPI, which is channel 1, and we're going to get we want to measure the intensity in channel 2. And so for this, in this particular case, we're actually going to put it in after selecting the ROI, and I'll explain that in a second. So essentially what we can do is go get our, from our previous macro, stack set channel 2, and we'll paste that in the loop. I'll just make a little bit more space here. After selecting the region of interest. Again, this is also on the GitHub if you'd rather copy paste it from there, and it's not critical to it's not so critical to running the macro just yet. The reason why I like setting the channel after selecting the ROI, so for each time this loop is going to go, it's going to select the ROI, so basically as though we're clicking on one of these things. And then after that, it's going to select the channel. It's that depending how you generate your ROIs, there might be a channel encoded within the region of interest. So this is not the case here, but if you generated your ROIs on a stack, for example, there would be a slice and a channel associated with that. So that would mean that if you were to click on it, it would go to the original channel. Thus, from there, you can select the next channel. So just kind of as to make your life easier downstream, since a region of interest can have X, Y, Z, T, and C information involved, then it's sometimes easier to get the ROI first and then set it to the slice that you want if you have to change it. But yeah. So let's unpack this a little bit. We have this for loop expression here at the top, which we'll break down. This is what we want to do over and over, right? So we want to select an ROI, and instead of giving it a number, I'm giving this iteration of this variable I, which is going to be what's going to change from one loop to the next. And so you can imagine it's going to go from 0 to num ROI and say, okay, ROI manager, select 0, select channel 2, run measure. Then it's going to do ROI manager 1, and so forth and so forth. So you can imagine how that works. To look a little bit into the sort of the mechanics of this, we got something like this. So this is our for loop, essentially. So for I equals 0, semicolon, I, the greatest integer under, the greatest integer smaller than num ROI, so the number of ROIs that we have, and I++, so adding I++, and this is the increment, so we're adding 1 at a time. It's kind of weird written like this. So if I can just kind of point this out like this, this means that the first part here, we're really just creating kind of a list of numbers, if you will, a sequence of numbers of integers, so no decimals. If we're creating a sequence of integers, then we have a start value, so we're going to start at 0. We have an end value, so we're going to take the greatest integer that's still smaller than num ROI, a variable that we defined earlier. And the increment from one number to the next is going to be 1. So this is a weird thing where I++ means an increment of 1. You could say I plus 2 if you want to do like 0, 2, 4, 6, 8, I plus 3 for 0, 3, 6, 9, etc. So if we just consider, hey, we're making a list of numbers, or the computer's making a list of numbers, then that's it. First, build a sequence of integers going from the start value to the end value with an increment of 1. So our ROI number is actually 5. I think we have 5 ROIs, so I'll just change that. When I was writing this, there were 6. So number of ROIs is the number of ROIs in our ROI manager. There are 5. So that means we're just creating, it's just under the hood, creating a list of numbers going from 0, our start value, to the greatest integer that's still smaller than our end value, which is going to be num ROI. Well, that's it. We're just creating that list of numbers. And then from that list of numbers, and again, whoops, we made this mistake here. once we create a list of numbers, we're essentially going to run the content of the loop, so everything that's between these curly braces, replacing I with the first integer of the sequence. So I here, the first time we're going to run it, is going to be 0. And then it's going to run that loop again, changing I for 1. And then it's going to run the loop again, changing I for 2, for 3, until the end of this list of numbers. So under the hood, basically the computer's creating this list of numbers and kind of doing something like this, where it's just saying, okay, I equals 0, do this. I equals 1, do this. I equals 2, do this. And essentially it's this variable that's changing all the time that's going to allow us to change the ROI from one to the next. Now notice this is kind of a cleaner way of doing it. And because we're defining our number of ROIs based on the current image, the active image that we just analyzed, that we just did the thresholding, then we will always get the proper number of ROIs. So if one image has only 2 nuclei, then it'll figure out that num ROI will be 2. It's going to do it twice. If another image has 10 ROIs, then num ROI will be equal to 10, and it's going to get all of them one after another. So this is sort of what the computer is doing under the hood. It's generating a sequence of integers from usually, and we usually just start at 0, going to a max value based on what we want. And then from there, doing the thing over and over. This might be a little bit unintuitive at first, and I totally get that. But it is a very commonly used expression. They're kind of fancier expressions now in other programming languages, but it is really, really useful to iterate through stuff. If you think of the batch processing that we did earlier with the first macro, it's actually doing a for loop under the hood. So basically it's getting the list of files in the folder, and then just opening the folder, applying our macro, and then closing it, going to the next image, and so forth. And so this is a very handy tool. If you don't fully grasp it just yet, don't worry about it. We'll run it, we'll see how it works. And if you do end up using it a few times and in different contexts, either for regions of interest or for selecting images, which we might have time to do right after, you'll start getting a feel for it a little bit, but it is unintuitive. So with that, we have something like this, which is pretty good. One last little decoration that I'll put in at the top of the script is that basically right now we're not running this on multiple files in a folder. We're just applying this on the open file, and we're not actually going to batch process this macro in this workshop, although you totally could work on it a little bit. So what we want to do is still make our lives a little bit easier. If we were to do this, to run this and then close this, open another file and run it, all these regions of interest from the previous file would still be there. So we'd find we'd end up with a new image, you know, something like this. And then we would still have the regions of interest from the previous cell, from the previous image, which is not ideal. So we want to put a command at the top of our macro to basically just delete the ROIs that are there once we open when we have an image. So for that, that's a very easy one. So I'll just go right after the set measurements and right before I say select, get image ID. We'll just type, you can start typing ROI manager. So this is an ROI manager thing. And then if you scroll down, you should find something called reset. So ROI manager. So ROI is lowercase manager with a capital M. And then in brackets and double quotes, reset with the regular brackets and the semicolon. And for ease, I'll just put it in the chat. And so that's going to delete that's going to get rid of the ROIs so it doesn't get confused with the nuclei or the ROIs of the previous image. So let's close some of these things. We don't need to save this. Close this, and I'm just going to get rid of this data here. And then if we just kind of start from scratch, if everything's there, if you kind of pasted everything in, see if this works. I'm going to choose one of my raw demo images. So I think we forget on which one we worked. I think it was 11. Let's just take number 1. And then if we say run, hopefully a bunch of stuff happens. And so it goes very quickly, so it's a little bit underwhelming. But then if you look at your ROI manager, you should have a bunch of ROIs. We can adjust the contrast of our images just to make sure that it makes sense. So again, that's with the brightness and contrast window, which is an image adjust brightness contrast. I'm just clicking auto. And then you can kind of scroll through and see what they look like, or you can click on the show all. And then somewhere in here, you should have a results table where it measured the intensity for all these 6 nuclei. Yes, 1, 2, 3, 4, 5, 6 in image number 1 here.
Magical.
Yes, I agree. So yeah, and there you have all these values that we chose earlier. So the mean intensity, the median, the raw sum value, the channel, which can be handy. And so then you can do this kind of over and over and kind of populate these results tables. This result table in Fiji, you can save it essentially as either an XLS file or a CSV file, which are data tables that you'll be able to open in Excel or in R or MATLAB or Python or what have you. Depending on the type of analysis you're doing, maybe you have one CSV file for your whole experiment. So, you know, we open the next image and the next values appear here for the next image or so forth. Maybe it makes more sense to save a CSV for each image. So you do file save as results for image 001. That's a little bit in the downstream processing that we're not going to discuss too much today. But here you get this kind of information here pretty quickly. And you can try it again. We can just close this stuff. We don't even have to close it, essentially. We can just open another image. So that was number 1. Let's do number 2. And make sure number 2 is selected. We say that. And we get those 2 cells there. And same thing, getting the intensity, within this phalloidin. And if we look at our results table, same thing. We have our the first nuclei from image 1 and the 2 nuclei from image 2, which indeed had 2 nuclei. And so the cool thing, I think, with the macros, that it does save you a bunch of time. You don't have, you know, if especially what you do automate, you do save a little bit of time. And you do end up having a slightly more structured readout. So if you're doing this entirely automatically, and we do have, for example, stack set channel 2 here in our for loop, then we're pretty certain it's always going to measure the second channel, the actin here. If you were to do this manually and you're doing this over and over and over, I can guarantee you that at some point you're going to accidentally measure the wrong channel. And that's a little bit frustrating because you'll end just because you'll get the wrong values. You'll be able hopefully to see it if you have your have the channel indicated in your results. And so that's what we did in the set measurements. That way you can go back, and if you do see a weird value, then, oh, I accidentally measured, you know, channel 3 or something. Actually, I can do that right now. Suppose we accidentally measured the mitochondria. You'd see this here, channel 3. So then you have to go back and fix it and this and that. So just having something a little bit more streamlined is very helpful. We have just a few minutes left. And so I would like to show you one last thing that will just kind of, since we don't have much time, we'll just kind of jump through somewhat quickly. I can stay around also on Zoom if there's other questions or concerns. But is everyone okay with seeing one last thing, or do you want to process this a little bit more? I can send you the PowerPoint instruction. Yeah, I mean, the PowerPoint is just what you saw today, which was just a few slides. But so there's just like the opening things and the breakdown of the for loop. The rest is on the GitHub. But yes, I can maybe just send me an email to remind me. Okay, so we'll do one last little thing. I'll say don't save. I'll close everything. And the idea is simply a little bit more real world to a certain example. So I'm still going to do it on these demo, on these toy images in a way. But it might apply a little bit, it might initially apply to your work a little bit more. And that's simply, you're going to get a bunch of images, and they may or may not look like this. But at some point, there might be things where it's actually difficult to do thresholding and segmentation. So we applied a median filter and an Otsu threshold, and we got the shape of the nuclei really nicely. And that's great. Maybe you're more interested in actin itself, and what is the intensity of, you know, this particular fiber here, or these stress fibers, or something like that. Or maybe you're interested in neurons, that the intensity within neurons, and the neurons are kind of close to each other. So it's difficult to get these separate structures. The DAPI images, they're very, these nuclei are very bright and they're spatially separate. They don't overlap. So it's very easy to find these objects. Getting the computer to find these individual filaments or cells that are very, very close to each other or touching is difficult. If I just apply a threshold here, like it's okay, not great. I get a bunch of noise and it's hard to determine what's the actual filament. So in cases such as this, it does happen that you'll have to do a little bit of manual analysis, and that's fine. That can be either because it's too difficult to write something, or you're just starting out, you're just getting images, and you just need to get a little bit of information, a little bit of data from, 10 cells or 20 cells. So to do that, maybe, I don't know, maybe you want to know what's the intensity of the actin fiber that's going, in the middle here, the one that's most in the middle, let's pretend. And maybe it's something else. Well, you're going to open up the image, you're going to go to channel 2, and maybe we're just going to draw a line, a freehand line, and you don't have to do this at the same time as me. This is just to kind of show you. And then you're going to draw along this fiber. You're going to do a better job than I do. And then maybe you do measure. So we get the measurements, the intensity along that fiber. And then if you're happy with that, you're going to close it, and then you're going to open the next image and kind of do that again. And then the same thing, you have to adjust the contrast, and then go to the second channel, adjust the contrast, and then draw that and click N and all that. So that's fine. It's just that it's tedious to do this on 20, 30, 40 images. Although we do have to understand that sometimes you still need to go in and do some sort of manual modification, or it requires your human scientist brain to decide. You can think of quality control as well. You want to make sure if your images are good. So rather than, you could at least use, even if you can't write a macro to do all the processing and find the objects for you, you can still use a macro to help you out and actually reduce some of the mindless clicking that we have to do. And so I'll just copy paste that kind of from the GitHub for now, since we're running low on time. So into the Fiji macros. I'll just do this here, exercise 3. And I'll just copy paste this whole thing. Oops, and I'll just try to move this window, which is proving to be difficult. All right, so paste that, ImageJ macro, and I'll just walk you through it. This is to help you, this is essentially a macro to help with manual analysis or quality control. And the idea is just write something that's gonna at least do some of the mindless clicking for you, which means opening the image, adjusting the contrast, and after you're done working on it, closing the image and opening the next one. And I can't stress how helpful this is, and how you can just get into your scientist mode of getting the data that you want, rather than having to open an image, adjust all the things yourself, do your measurement, close it, and you end up opening the same image again by accident, and the data is frustrating and all that. So this is exercise 3, just to kind of walk through it. Essentially what we're going to do is that we're going to use a for loop, which you may have seen before. Here I'm using the iterator variable k instead of I. That's just, I would do the same thing. K is just handy because if I do another for loop inside this, they don't kind of crosstalk and do something nasty. And essentially what we do, instead of going through these regions of interest, we're going to go through a folder of images. So I have a command here called get directory. And this opens a window for user to select a folder. That's going to store it under the variable that I just called input. Then from that folder, which is just going to be, C:/ Joel's computer / desktop / demo images, we can then go get the list of files in that. So get file list from input. And again, this is just going to be blocks of stuff that you're going to paste yourself. And then for a for loop, we need the number of iterations that we want to go through, right? So when we had the ROIs, we needed to figure out how many ROIs we had to loop through. So this is the same thing for the files. We need to get the length of the list of files. How many files are in there? And then we're going to write our for loop, and so that's for k = 0, greatest integer that is still smaller than the number of files, and incrementing 1. We have this command here that says open input. I'll just add some spaces to make it clear. So input, which is the folder, so C desktop demo images. And then list of files with the value at the position K. So k is our iterator value here, which means the first time we run this, k will be equal to 0. 0 will be the first element in that list of files. So it'll be the first file. And then it's going to do it again, k equals 1. So list of files 1, that's going to be the second, and so forth. So it's going to open this image. I'm going to get the title, because that's kind of cool. You can store the file name, and I'm going to print it out. And maybe you want to do that, maybe you don't. I'm going to adjust the contrast, similar as we did in our first macro. So I'll say stack set channel to 1, enhance contrast, 2, enhance contrast, 3, enhance contrast. So that means I don't have to go in and click auto on each of them. It's going to do it automatically. And then this is kind of where the magic happens in this particular scenario, is that we have we generate a wait for user window. And this is a window that will just say whatever I want it to say. So make a measurement or check for quality control, then click OK. But it's going to allow me to access all the things within the macro. And then it'll allow me to make a measurement. And then once I'm done with that, I click OK, and it's going to close automatically. Now, this might seem really, really silly, but it really makes your life easier if you have to do manual analysis for quality control. Like you just want to see if your images are good. So I'll just click Run to give you an example. So as I said, at first asks you for a folder of images. So choose a source folder. We'll choose demo images. We'll say OK. And then printed out in the log the file name, which is handy. So right now I know that I'm working on 001, and that's handy because sometimes you have a really long file name. It's easier to read here. We have this window here, say action required, that's waiting for me to click OK. And in the meantime, I have access to everything. I can kind of do whatever I want. So then here, I just clicked run once. It opened the image, it adjusted the contrast. Let's go in, let's make a measurement. So let's say we want to get the information from this filament. I'll say M to measure. Just make sure that I actually measured it. Start again. So we'll say M to measure. And let's say that's all I want from this image. I say OK. And then it opened the next image and again, adjust the contrast for all of these. So then I can go in, use my, really do more the scientist job as opposed to the monkey clicking job. Let's make a measurement here. Say M. Maybe there's another filament here that I'm interested in. Let's say this one. Say M, and then you just say OK. And so it's kind of doing a good bit of the boring work for you. It's always going to open the next image. It's never going to open the same image twice, and just kind of goes through and makes the process a lot more streamlined, a lot easier to undertake. It might seem silly at first, but if you do have to draw things or make measurements, or even just say, oh, is this image good or not? Well, you can just run through them and either make a measurement if it's good, or write a little note somewhere, oh, image 2 is bad, image 13 is good, something like that. And so you're just clicking OK. This wait for user just gives you a pause to go in and do something. Maybe this one, we'll say OK. Nothing good here. Well, this one's kind of funny. It's OK. And yeah. And it'll just go through the whole folder. Where are we now? Almost done. There we go. And so this is really, really handy. It should be in the GitHub. It's so it's not in the README. So just to finish on this, I realize people have to leave, and I'll stop there. This can be really, really handy. You can add pretty much any sort of processing you want. So enhance the contrast, set it to a certain channel, maybe a filter, and then with the wait for user, you kind of do whatever you need to do. Either just look at it, make a decision, measure something that's difficult to automate, and then you just click OK, and it just runs to the next one. So I really, really recommend getting used to this, because it will make your life a lot easier, even if you're just looking at your images to get an idea what they look like. So I'll stop there. I'll try to remember to send you the PowerPoint. If I don't, please let me know. This particular macro, it's not in the README of the GitHub. So if you open the GitHub page, so again, just intro to Fiji macros, rather than scrolling down, there's a list of files here. So there's exercise 1, 2, and 3. If you click on 3, you can either download it, which is totally fine, or you can also just copy paste it directly. It's not going to copy the number lines on the side here. And this we're going to leave on, indefinitely. So you'll have access to this pretty much any time. So it should be there. Yeah. All right, so I will stop there. It has been 2 hours. It's for some of you, this was super introductory. I'd be curious to know if you do have experience writing either in other languages or in this, or in Fiji, and I know some of you have. I've seen you. Let me know what you think about this, if this is kind of how you would have taught it differently, or there are glaring things that are missing. For those of you that are sort of a little bit more new to this, please give it a shot. What we tried to do is really keep it on the sort of practical side of manipulating images, and not just, you know, starting with, oh, a variable can be 2 and 2 + 2 = 4 and stuff like that, which is really cool for computer science type approaches, but I think we sort of want to get you going with modifying images as soon as possible. And through that, if this is interesting, then you'll sort of develop kind of a sense for it, and kind of figure out where you can be tricky, where you can be clever to get sort of convince the computer to give you the information that you want. So I'll stop there. Thank you so much for coming. Thank you so much for joining in large numbers. And if possible, can we get a picture with people?